Cells vary in size and shape. Let's have one example of the kind of cells that we can observe under microscope. Appear to be polygonal cells, and these are actually flat cells of the skin. We also have these cells in this flat form, but our skin cells are being lost daily, and so it is not easy to collect those skin cells. We can collect some of the cells from our cheek inside. Using the glass slide carefully, it is possible to scrape a little bit of the inner part of the cheeks, but that's not very healthy in one sense, because there is a possibility of injury. But then, there are animals like frog who lose their skin in water, and by this way they renew their body cover, and that's happening, and all of us are losing our skin. But the frogs will lose the skin in full, and such skin pieces can be observed after staining. I mean, that's a natural process, and you can collect it from the field, and the staining may be done, and the cells will be observed as squamous epithelium. We are going to learn this separate chapter, epithelial tissue, and that time we are going to learn this word squamous. What is squamous epithelium? They are flat, like flooring of tiles, the flooring of our rooms, the tiles, and they are flat like the flooring of tiles, and this is all therefore called as pavement epithelium. Don't worry about epithelium, that word, and the different tissues. Just have an understanding there that this is one way we can see those cells. Let's make a slide and let us observe it now. Let's now make a slide for the skin epithelium. Look here, you will find here a very small piece of the skin. In fact, you don't need so much, you require a very small piece. Okay, we have this piece available with us, and this will have many thousands of cells, remember. And I'll try to this was originally in formalin. Now I'm putting it in water. It's being washed. So what is the first step of making a slide? Washing. After washing, you go for staining. Here is a stain, and this stain is eosin stain. What is the name of the stain? Eosin. And eosin is a acidic stain. It is acidic stain, means it will stain the basic things in the cell. There are two types of stains, acidic and basic. I'll tell you one example of a basic stain, hematoxylin. It's a basic stain. Or acetocarmine, it's a basic stain. So basic stain will stain acidic things. When the nucleus is to be stained, you have to use a basic stain, because nucleus is acidic, there is nucleic acid, and therefore you require a basic stain. So acetocarmine is basic and hematoxylin is basic, and they will stain nucleus specifically. Eosin is an acidic stain, and eosin will stain the basic compounds in the cytoplasm. Usually it's a generalized stain. So I'm just taking a few drops of the eosin here in this watch glass. I think shake it and shake it. Okay, so I'll just put back the lid and keep it away. I'll just put this skin piece here. I put this in stain. I'll just put it for a few minutes, and then I'll again give a water wash, and then let's see what happens. See what is happening now. Already it is a preserved thing. Preservation is kind of fixation. I'm introducing to you a term, fixation. Not all tissues will require fixation, but the animal tissues usually will require fixation. So when the things are kept in acetic acid or alcohol or formalin or a mixture of these things, which three things I have said? Acetic acid, formalin, alcohol, you can make possible mixtures of this and put it there. It was already kept there for years, it has been there, and it is now fixed. By fixation, what means is the proteins inside the cells, they get stabilized, and then staining is possible. If you take a fresh piece of the things, if you take the fresh skin and then try to stain, the staining will not be so good, because the proteins are unstable, they are still scattered. You want the proteins to have their positions fixed, and then they should be then and there, so that the stain will go and catch them. So proteins and many other materials get fixed, so fixation is very important. There's a lot of theory about how the tissues are to be fixed. Now because this is fixed, it can be better stained. Without fixing, the staining may be weak, don't worry much about it. So after staining, and it's a matter of convenience and experience, how much time to allow for the fixing. In my opinion, whatever 2 minutes have been there may be generally good enough, and I'll just put it for washing. What is this second step, washing? Washing is to remove the extra stain. Can you see lot of extra stain is being removed? And in fact, I'm going to do the washing for more times. I'll take some more water in these wash glasses, and then I will try to shift this skin piece there, and you'll find that all extra stain is being removed. Extra stain is being removed. In fact, I can take some more water and allow the removal of the stain. Okay, watch here. I think not much of the stain is, it seems that now the extra stain has been removed. That's very fine. So now you can see this is now stained fine, and I'll take a small piece. You can make 5-10 slides out of this piece. You require a very small piece. I'll use the scissors to take a very small piece. This is the only scissor available. It's okay, fine. I'm now going to take a drop of water here. I think I'll still use the white background, that is still better. Okay, now I'll take a drop of water. I'll just take a tiny drop of water here, and I think I can use a better scissors. Yeah, I'll take this one. Then I'll pick up this piece of skin and cut it into pieces, and I require only a small piece, a tiny piece. With this small tiny piece is good enough. In fact, I'm going to choose that only first. Can you see this tiny piece? It's good enough. It's quite good. You'll have many thousands of cells there. Okay, I'll take one more small piece. Oh my God, there are so many small pieces, and I can take a small piece here. It's very nice work, but for biology practical, you should have a kind of a delicate hand, they say. It should not be harsh, and okay, I'll take this piece, whatever it is. I'm taking it here. See, almost 3 pieces here. Fine. I think I can just put a drop of water additionally, and then put a cover glass. Sure. This is a box which contains cover glass. Most of you know, but some of you probably are looking at these things for the first time. Can you see? They're very thin, very delicate, and you have to hold them by their sides. Why? Because if you hold them from their surfaces, then your fingerprints will be appearing on them. So it is good to hold them, hold the cover glass from a slide. Okay. Oh my God, I'm just I'll put them here, and I'll just try to pick up only one of them. These are actually 2. So okay, they are stuck to each other. I think I'll pick up a simple one, a thin one. Yeah, here I can see. Can you see this one? This is very thin. I'll put these 2 back in the position. So you can see this one single one. And now what I'll do is I'll try to put this here. Now for that, it is good to support the cover glass with the help of a kind of simple needle, and then very slowly, see, very slowly I'm trying to drop the cover glass on the material taken. See, that's it. And now that extra water can be removed. Just put a piece of paper, a tissue paper, a blotting paper. Allow some water to remain, but all extra should be removed. You know what is important for biological work is cleanliness. If the cleanliness is there, then the material that you are likely to observe will be seen in a definitely good way, because there should not be a mess which you're making. Slide making becomes an art, and you have to do it in a very delicate manner. Just blot away the extra liquid around, and it should be as clean as possible, as clean as possible. Particularly it should not be wet from underside, because when you put it onto the stage of the microscope, it should not get stuck to that. So this is the slide ready now. Let's now go for adjusting the things under microscope. So here is the slide that we have just now prepared. I will now adjust it under the compound microscope. I think you are already, most of you are well aware of the parts of the microscope. This is the base, and this is the platform on which the object is placed, and this is a lens here which is called as objective lens, and there is a lens which is closer to the object, and so these are objective lenses. As you can see, I can move this. This is what is called as nose piece, and it can be moved. So the major parts are the eyepiece lens, which is called as ocular lens, the objective lens, which is close to the objective. This is a stage, and it can be moved. Can you see this? It can be moved. It's possible to fix the slide in this clip, and it can be moved. There are two important adjustments. This is called as coarse adjustment. Look here, the things. The coarse adjustment is for a rough adjustment, you can say, for bringing the object in focus. But if you want to make the final adjustment, you have to use another screw here, this one, which is called as fine adjustment. Once again, this is coarse adjustment. You can very easily appreciate the movement of the stage. But this is fine adjustment. Even through this, the stage is moving, but it's moving through very small distance, through millimeters, and so you are not able to perceive it easily, but it is moving. That's fine adjustment. This is what is called as this piece here down below is what is called as the condenser. Here is a condenser. What is it? It's condenser. And that's a lens again, and there is a diaphragm for the condenser. Can you see this knob? This is a diaphragm for the condenser. All this is to adjust the light. So once again, you have the coarse adjustment, this is fine adjustment, this is condenser lens to condense the light, and the diaphragm to regulate the amount of light that is passing. These are the main things, and now let us adjust the slide. I'll adjust the slide. Okay, I'll bring it in focus first. Adjustment will require a little practice, and many times it takes more time. That is also normal. Some of you might have handled the microscopes in your schools, and once or maybe that you have your own microscopes at home. If you are medical field, probably microscope is more likely to be there. The things.
Oh, the slide is coming up nicely. Oh, beautiful. It's really good comparatively. Okay, fine things.
But of course, we'll click a picture. It's possible for us to just put our camera of the handset here. It takes time, of course. We'll click a picture and then show it to you. Okay, let's go for that. Look at this. A beautiful slide has come up, and fortunately we could picture it nicely. Can you see the cells now, which are having many sides and they are flat, and you can see the nucleus even here. That's how, very simple procedure. But then it is so much of informative, educative, and most important, it provides us so much of pleasure. So why do we learn science? Why do we learn biology? Why do we learn physics and chemistry? It's for pleasure. It's for the kind of satisfaction that we get out of it. NEET and all are very small things. You are not studying biology, physics, chemistry for NEET. You are studying science for being enlightened, and that's more important. Well, so with simple arrangements here, we could make a slide and show it to you, thanks to those people who generated laptop and the microscope, and most important, the device, the mobile piece, and so on. That is what is science. Through internet, such a effect, such a picture could be obtained. Naturally, this is stained differently. Here, the nuclear stain has also been used, and nuclear stain is something which is basically a basic stain, because nucleus has acidic compound, nucleic acid, and then it gets better stain with a basic stain. So once again, going back, this is our slide here, which is differently stained. This we have used the eosin, which is a cytoplasmic stain, and this is a basic stain. So that is it, and I'm sure you love to do this virtually. To say, this is something which is happening all over the world. Now schools and colleges and classes have stopped, so it's okay. Still, I think it's good to enjoy the things that is at hand and that is available. Okay, so we'll continue with the next item of the practical now. So our next item for the practical demonstration is to make a blood smear to observe the red cells. White cells also can be observed, but there are certain issues which I am going to explain. I think today we will be restricting ourselves to the red cells. For that to happen, we have to prepare a smear. We have to have a drop of blood first. So on a given slide, we will have a drop of blood, and with the second slide, I'm just going to spread the drop of blood. You may wonder whether it is really very difficult. To be very frank, it is not difficult at all. It's quite easy, provided a drop of blood is available. Well, I'm sure someone of you who are observing the video will be able to come here and give a drop of blood on the slide. If not, let's have some alternative. The blood can be taken out with the help of blood lancets. Here is a box where you have many lancets. Can you see? This is a box full of lancets, and here I can just pour a few lancets here for your observation. What is a lancet? It is a very fine tip, a needle which has been fitted here. I can show you. For example, I'm just pulling it. This is the one, very fine, and it is this one which can be used to puncture the finger to take out the drop of blood. Well, it's already sterilized, so there is no problem, and I'm putting back the others just to make the stage clear for demo. Now, sterilization is very important, because you have to sterilize the finger and then just take a drop of blood. That's quite easy. Let me tell you, suppose a drop of blood has been taken, then the way I am going to do it. So, suppose the drop of blood is here. Then I am just going to see if the drop of blood is here. just a few millimeter away from the drop of blood, I'm just going to hold the spreader. This slide will be called as a spreader. I'm going to have a spread here and it's a spreader. So I'm just, here is a drop of blood, I'm just going to hold the spreader a few millimeters away from the drop. And then I'm just going to move it towards the drop. And then quickly move back. I'll repeat. A drop of blood is taken here, a tiny drop of blood, a tiny, not very big. Then a few millimeters behind the drop of blood, I'm just holding the edge of the spreader. And then I'm just moving it to the blood drop. And then quickly, with enough pressure, I'm just only once moving it backward. And the blood will be spread. Interestingly, the blood gets dried within a few minutes, the smear, and then it can be directly observed under a microscope. You don't require any kind of cover glass to be put in. That's one thing. Secondly, you don't need any staining also. Staining is required. Okay, I'll say about the staining. Here is what is called as Leishman stain. Can you read this? Leishman stain. This Leishman stain is meant for observation of the WBCs. Today, I think we are not going to do it because we have many other items in the sequence. But Leishman staining will definitely be shown to you once we go for the chapter of circulation sometime later on, the blood and other things. But then let me say a few things about this particular method. Leishman was a great person in the British Army. He was a medical professional in the army and he was a pathologist. So naturally he had a background of the military as well as medical science. And then he's the one who started around 1900. More than 120 years back, he started with this particular procedure and it is very much used even now. Here is one slide which I can show you. Here is one slide. Can you see this particular stain? This is purple colored. This is the slide which already has been made here using Leishman stain. We can show this to you, okay. But actual steps of the Leishman staining we are not going to show you. So what we are going to do, we are just helping you to observe the making of a smear. And this directly it can be put under the microscope and you can observe the red cells. Then this prepared side of the blood smear using the Leishman stain will also be put under the microscope and you can see white blood cells. White blood cells will be observed only if the staining is done. But for red cells, you don't require any staining. And one more thing, when you are going to observe the red cells, don't expect them to look red. When you are going to observe the red blood cells, erythrocytes, don't expect them to be seen as red. Only when they are in together, clustered, then only the red color appears. And that's about the color theory. Every red blood cell is very tiny. Think of the different shapes possible. It's about 7 to 8 microns. And you know, speciality of the RBC is that it is circular, not spherical, circular and biconcave. Human, mammalian RBCs are circular and biconcave. Under microscope, you may not be able to observe the concavity. You may mistake the concavity as a nucleus because you know that concavity will provide a kind of a shade in the image, and then you may mistake that as to be a nucleus. But it is no, it's wrong because the RBCs, mammalian RBCs, human red blood cells do not have a nucleus, that you know very well. Every red blood cell is packed with hemoglobin. Every single RBC, which is very, very tiny, is packed with hemoglobin. Let me tell you the estimated number, some 280 million hemoglobin molecules. A tiny single red blood cell has 280 million hemoglobin molecules, and that is how it has the power to carry the oxygen. And you know already that one hemoglobin molecule carries 4 molecules of oxygen. One hemoglobin molecule can carry 4 molecules of oxygen. So Hb4O2. The formula becomes Hb is hemoglobin 4O2.
and suppose there are 280 million hemoglobin molecule, and every hemoglobin molecule suppose carries 4 oxygen molecule. You can imagine 280 million into 4. Those many oxygen molecules will be transported by a single red blood cell. It's enormous. I think the body machinery, the design, the mechanisms are many times beyond the human ability to understand and appreciate. So look at these things from this angle. Okay, I think we'll go for making of the slide now. I may request one of you to come forward, please. Many of you are listening. Why don't you come forward? Yeah, please come forward. Yeah, here we have, you know him very well, Kaustubh sir. Say something.
I'm here to demonstrate the blood smear experiment.
And are you afraid of blood?
I'm not afraid of it.
Okay. So I think we'll proceed. I think this is our regular sanitizer, alcohol, whatever is available. I think, okay, as usual. This is very important these days. Otherwise also, you have to definitely sterilize your finger. So I think, so you can have a, yeah, please do it, yes. I think we need a cotton swab also. Can you just get it?
Yes. Tissue.
A tissue or cotton swab. He is bringing that. And meantime, let me tell you that it is very important to discard the needle. That's very important because earlier there used to be times, old times, 25, 30, 40 years back, that the same needle used to be used for many people. Not only for this kind of blood collection, also for any kind of injection. People when they get injections, yeah, please be here. Then they used to use the same type of needle. They used to boil it at the most. Nowadays, of course, that's not possible. That never should be done because there are lots of possibilities of transfer of diseases through the blood, and so it has to be very carefully done. I can now use this very tiny cotton swab. I'm just going to clean it here. See, are you feeling good, right?
Yes.
Oh, that's it. Alcohol. That's nice. Something. Okay, let it be dried, and I'll just put this tiny thing into your finger.
Sure.
And you'll not even know about it. Okay, and I'm going to take that small drop of blood here on the slide here. Okay? Are you comfortable?
I'm definitely.
When actual practicals we conduct here, now because of lockdown that is not possible, it's very interesting. Students come in a group and one of them is then asked to volunteer for a drop of blood. And yeah, somebody comes forward, there's no doubt. And there are some people who would keep away and then they say, oh, no. Even, why the drop of blood generates why it generates fear?
It is a psychological. Someone
really? Okay.
Afraid of it.
Okay, I have pricked his finger and I'm sure some blood is going to come out. See this drop of blood. Oh, it's good. It's enough. Just put some. Okay. That's very fine. Have, I think, another cotton piece to sterilize once again, because you need to sterilize the things. Okay, it's very simple.
Yes.
You did not find any pains, right?
No.
Okay, and I'm discarding this, very important. I'm just discarding it. You don't have to use it again. I will take a spreader now. Suppose this is a spreader slide.
I'll go behind the camera.
Oh, you don't need to go to behind the camera. Okay, fine, thank you. So that is how we work. Somebody is behind the camera, somebody is in front of the camera, and we wish that sometimes one of you may also join us either in front of the camera or behind the camera. So here is a drop of blood. Now see what I am doing. I am trying to now put this spreader a few millimeters away from the drop of blood, and see the way I'm holding it. I'm just putting it a few millimeters away from the drop of blood, and as I showed you, I'm just moving it up and then quickly behind. Oh, not a very good spread, but I think it's okay. When you want to observe this under a microscope, preferably try to observe the edges, because there are two things. The edges of a smear will have more thin layer of the blood. That's one thing. So you can observe the cells better. And secondly, when such a process is done, ultimately blood is a living tissue. a fluid connective tissue, you know this. And the white blood cells will try to migrate to the sides. So when Leishman staining is done, this seems to be done with a bigger drop of blood, of course. When Leishman staining is done, you can expect that the cells, if you want to observe the white blood cells, they can probably be observed on the surrounding areas. Well, this is the spread that we have done, and we can then arrange this under microscope, and you can see this one which is freshly done, and also you will be able to observe the prepared slide with the white blood cells. This is what we will do, and this is a very simple way, because our purpose today is simple. We want to know the different types of cells. I think you have already observed a few minutes back about the epithelial cells. A very nice slide could come up, the skin cells, and this is what we are going to observe now. I'm just allowing this to get air dried. You know, it can be air dried automatically. Then after this, I'm going to go for what are called as raphides. Okay, but about that I'll talk little later. Let's now go for making this slide adjusted under microscope. So here is the slide ready now, and I'm just putting it onto the stage with the slide holder which is in place. I'm sorry for this. So I'm putting this here. I'm not having any cover slip done. That's it. And then the light adjustment. Try to select the area which has got, first there should be low power. Yeah, select the area which has got lesser cells. I mean, there's less crowding of the cells, where the smear is thin. Usually at the edges the smear is thin. So here it is, smear is thin, and yeah, it is possible to adjust. You can look at me very carefully. I'm keeping both eyes open. That's very, very important. You should put both your hands together on the adjustment, the coarse or the fine, and keep both eyes open. असं बऱ्याच लोकांना वाटतं की एका डोळ्याने एक डोळा बंद करून बघायचं असतं. असं काही नसतं. It's all it's a wrong way. Many people are wrongly trained. It has never to be seen. See, I'm looking through both eyes open. What is the advantage of keeping both eyes open? I can look for a longer time. Why? Because if I keep one eye shut, then the eye that is shut gets some strain, and the eye that is open also gets some strain. While I am keeping both eyes open, look at me, and I can see very comfortably. So whenever you get a chance to look through the microscope, right from beginning you make a practice to observe with both eyes open. Okay. Now, Kaustubh sir, can you just take a picture? This is a condenser. I'll adjust the condenser also. Look at this condenser. Yesterday, in the earlier practical also, you have seen the movement of the condenser. So that condenser lens is moving up and down. It's fine. Just take a click, please.
So now I'm just looking, and I'll find an area where the cells are not much in density, and yes, I found the spot, and now I'll click a picture and I'll just show it to you. I've adjusted this.
So you can make the zoom with the help of the microscope plus the camera. Okay.
Yes, definitely. So yes, I found a great spot. I'll focus a little bit. Fine adjustment. I'm using the fine adjustment, and after I focused it well, I've clicked a couple of pictures now, and yes. So yes, I've got a couple of pictures, beautiful pictures, and I'll be showing it to you now.
It's very interesting that it's good to be behind the camera sometimes. I would like to be. Okay.
Definitely.
So here are the results. You can see two slides here. That's mere two slides. One we have prepared just now, which is not stained, and only red blood cells will be observed, and we are going to show the picture that we have clicked here. So there only RBCs are observed. On the other hand, on the right hand side, you have a purple colored smear, because that smear has been stained with Leishman stain. Leishman, we are going to show you this great person, the picture of this great person, where you will find the smartness of this military personality who was a pathologist, medical professional in the military, and then he was the one who around 1900 tried to invent a Leishman staining, and the staining, and then it was being called as Leishman stain now, and for the more than 100 years it is being used everywhere. Here you can observe both red blood cells and WBCs. Even this has been pictured, and you will be shown that shortly. This is the great person, William Boog Leishman, Scottish pathologist and British Army medical officer, who described a method of staining blood for malaria and other parasites in 1900. He modified an existing method using a compound of methylene blue. Yeah, the stain is methylene blue, and that's the methylene blue is a basic stain, and eosin is an acidic stain, which is now known as Leishman stain. We go ahead. These are the pictures that we have taken. Interestingly, both are red blood cells, but with different light effects. They are unstained from that unstained sphere, and you will not be able to observe clearly any WBCs. Only red blood cells are seen, and usually you think that RBCs are circular. True, they are circular, but because they have been taken out, I think some of them may be slightly undergone change in shape, so they may not be seen exactly circular. That's quite possible. And interestingly, the red cells are not looking red. You can ask a question to yourself and think why. In fact, if you take a drop of red watercolor and spread it on a big sheet of paper, what I say, take a drop of red watercolor and spread it onto entire white sheet with a brush, and the red color will vanish. Then collect all that thing once again into a drop, if you can do that, and you'll get the red color. So this is something which is great, and you can think of the physics behind this color theory, and think why this is happening. So individual red cells are not red, but they are put together in a cluster. They appear to be red. Here is the stained sphere. We did not stain it just now. It was stained earlier. You can see very clearly the cells which are all here are all the red blood cells, which are now actually kind of clumped together, as you can see them. But the two white blood cells are very clearly observed. You can look at this. This is neutrophil, and the neutrophil has multilobe nucleus, because when we learnt the nucleus, we commented on the nucleus can be of different shapes. Look at the nucleus here. It is spherical nucleus almost. And so this is lymphocyte, and is a neutrophil. So after staining, the red blood cells seem to have, maybe that the stain, when the smear was taken, that time the cells might have got clumped together. So this is the result of the Leishman staining. So Leishman staining is more specific. You can differentiate. You can differentiate RBCs from WBCs. So the purpose of the staining is that to differentiate. That means to distinguish one type of cell from another type of cell. And you can expect then why this nucleus has become, why it has taken more stain. Some stain has gone here into the cytoplasm also. But what has gone into the cytoplasm is eosin. And eosin is an acidic stain. And methylene blue, ultimately the Leishman's stain is a mixture of methylene blue and eosin. And methylene blue is a basic stain which goes and attaches to the nucleic acid in the nucleus, because nucleic acids are acidic and the methylene blue is basic, so they come together, and that provides the darkness. This is the slide, this is the picture collected from the internet. So of course you can expect that the slide that we are making has got some limitations, but this is the picture from the net. There are better fine methods to do the same thing. Now we will stop talking about the blood and its cells. We'll go ahead with the next thing, that is we are going to observe now raphides.
See, now we are going to take into account certain things which are stored in plant cells, particularly parenchyma. This is amorphophallus, alu, as you know very well. And in many such plants, you get what are called as raphides.
Raphites are needle-like structures, and they are actually crystalline, and they are crystals of either calcium oxalate or calcium carbonate. They are in the parenchyma cells, mostly in the petiole of the leaf, mainly. This is a very important family, and this is something which is quite edible, and we make a lot of preparations out of this. But anytime you make preparation out of this leaf, you are supposed to use something like tamarind chintz. Why? Because these crystals, they cause itching, and because they are pointed, so it is good to dissolve them. And so the tamarind chintz is being used for any preparation which you usually make out of this arum alu. This family is Araceae. Now you are learning classification, taxonomy, and there are plants divided into families, more than 300 families, and Araceae is one of the families. Actually, Raphites had many such excretory substances. I can call this an excretory substance because it is in usually whenever the calcium is in excess, these crystals are formed. And many plant families will have either Raphites or something like cystolith. I'll just write one word. Cystolith. And cystolith you get in the ficus. We will just show you the picture of the cystolith. So it seems that the excretory wastes of the these can be called as excretory wastes. Sometimes these are considered to be a mechanism against herbivores because when these are consumed, the herbivores probably also must be having some problem, some kind of itching because of these crystals. So lots of things are there. But it will be good to observe. What we will do is we will take a section of this petiole, because this is the stalk of the leaf, and then we will see parenchyma cells, and in the parenchyma cells we will be able to see the Raphites. And in the case of the petals, the parenchyma also has big vacuoles which has pigments like anthocyanin, and similarly many of the parenchyma cells are quite possibly storing some excretory waste. Now to call this as excretory waste will be objectionable by many of you, but usually they are classified as excretory wastes. Let's go for making the slide and observing it under microscope. Raphites. We can obtain Raphites from many parts of the leaf or from the petiole. I would prefer to do it from the petiole. A very simple thing I'll do. I'll just take a piece of the petiole. It's very interesting. There is a lot of parenchyma. It's here there is a lot of soft tissue. So air-filled parenchyma. I mean, the surrounding of the parenchyma has a lot of air cavities. So let's now go for observation of the things, but I have to take sections. So I'll just put it back here, and now I'll take a few sections in the watch glass as usual. I'll take, usually you take a few drops of water. And yes, that should be enough. I'll take some sections. Okay, I think I can select this one and I can make a slide out of it. They can be still more thinner, but it should be okay. I'll just take a drop of water here and then put on the drop of water this section. This is what I am trying to put here. Can you observe this? And I'll put a cover glass. I'm just putting a cover glass here. I'll lightly tap it some water. I think I like to change the section because I think it is not uniform, so I don't think it is possible to do it. So I'll take another section and once again repeat the things. Once again I'll take the section. I mean, the thinner the section, the better it is. Oh, the small piece is really good enough. Can you see this very small piece, the tiny piece? Such a small piece is usually good enough. I'll now once again use the same slide, but I'll clean it. You know, cleaning is very important. Everything should be clean, neat, tidy, so that the final slide is likely to appear good. So I'll put a drop of water once again, just a drop of water, and I'll put this particular piece of this is the one, I'm just keeping it here, and now I'll put a cover glass, and this single cover glass I'll put, I think they are double, this is good, now this is single. I'll put a cover glass here, look here, look carefully, yeah, I'm dropping it. Lightly I will tap it, and there's a small gap left, and I'll fill it up with water. I think that is done. I think I can adjust this slide now under the microscope as usual. No staining is required. You don't think that every time you require staining. That's not really the need. Staining is required only if you want to differentiate things. And many times the slides made like this are more than enough. You don't need to always stain the slide. I will now put it under microscope. So I am now trying to put this slide under the low power of the microscope because I think the cells are going to be quite large here. So I'll put this on, and let's now adjust. No, I can see very beautiful air spaces. You know, this is a monocot family, and I think you are aware that this plant grows usually when there is ample water supply. I can see lots and lots of parenchyma cells, and there are some air-filled spaces, parenchyma therefore. Oh my God, I got the raphides. Some of the air spaces here are full of raphides. I told you on the board that it is the parenchyma cells which have the raphides, but then there are raphides also stored into the spaces. It's very, very beautiful. You can see those air spaces, air-filled spaces in which the raphides are there, and there are raphides also inside the cells, and many good things. I think it will be good. We will load this picture onto the computer now. So here are two pictures. This is one that has been taken from the midrib. We did not show that to you, of course. And this is the one that is taken from the petiole. Just now the slide was made. You can see here the raphides in bundles, and here you can see the raphides in group. But you can see some of the raphides are coming into a bundle form here. So these are the raphides, and but then this is not a see, these are all parenchyma cells around. These are all parenchyma cells around, as you can see the parenchyma cells here also. But then this is it's a space, air-filled space, parenchyma, so air-filled space which is being bordered by the parenchyma, and it is therefore the raphides being stored into the space. That is also possible, and there will be raphides also inside the cells. That's not being seen here. A very beautiful picture. So this is how there can be some non-living material. You can call it as cell inclusions which are there, and now you can expect that this is being it's being given out of the cells, and then probably it will be given out. But you know, in plants nothing can be given out. You know, ultimately it will lie there, and till the end of the plant it will be there, and I don't think it's going to cause any toxicity, but it's quite interesting. So we will be able to see one such picture of the raphides and cystoliths, and we'll show it to you afterwards. Here is one such way of storing the extra mineral in the leaves. This is the banyan leaf. The Ficus bengalensis is the scientific name. You are learning scientific names in this chapter. Ficus bengalensis, banyan, Vadacha झाड. So there you can observe here these structures. So these are all enlarged cells of the epidermis, you can say, and then you can observe here a lot of crystalline matter which is accumulated. So that's how things. Of course, then what happens in plants is these leaves then fall off. So the concept of excretion in plants is different from that in the animals. Let's now go for one more picture, and these are some of the rhaphides and spirophytes. You can read this later on. You see, these are the C, for example, C rhaphides in the petiole of Colocasia. Amorphophallus and Colocasia are common plants. So you can see here bundles. Okay, these are spirophytes. Spirophytes are crystals which are all put together in a mosaic form. It becomes a kind of a cluster, and there can be many such things. So that's how some of the cells. This is to show you that yes, cells can store non-living material, which you can call as inclusions, and many of them may be the excretory wastes.